Hi guys, this is Walker and my partner Esteban, and we're going to be discussing the effects of adding adenosine on an elevated Daphnia magna heart rate. First off, we'd like to give you guys a little introduction about what organism we used. So we decided to use the Daphnia magna, which is a common water flea, and they're small crustaceans that grow to about 3 millimeters in length or less than that. And they're typically used as food for other animals like fish, tadpoles, newts, etc. But the main reason why we chose them for our experiment is that they're translucent, so you can clearly see all of their organs under a microscope. And this allowed us to clearly identify their heart and be able to measure beats per minute. So one of the things that we consulted before deciding what to do for our experiment was a past experiment done by Aman Kundu and Ganesh Singh. And basically they studied adding caffeine into a solution and then adding Daphnia into that same solution and noticing how their heart rate changed after leaving them in there for a short amount of time. And this is the graph that they presented once they had done their experiment with plenty of trials. You can clearly see that the heart rate does become elevated in the Daphnia magna once you add caffeine. Next slide. Just briefly to let you know how caffeine works. Basically in our body, there are things called adenosine receptors, which take in a chemical compound called adenosine. And this job is to make humans tired. Now, adenosine has a certain shape and caffeine has that exact same shape. So caffeine can kind of sneak in to the adenosine receptor posing as an adenosine compound, and it blocks the adenosine from getting in there, AKA inhibits the humans from actually feeling tired. So that's the way caffeine works, and it works the exact same way on a Daphnia magna. Just to give you our hypothesis. So when we first started this experiment, we predicted that the Daphnia magna's already high heart rate due to ingested caffeine will decrease with greater amounts of adenosine. And we said that that is because if more adenosine is added, then it will competitively interfere with the caffeine at an increased rate, and it will bind with the A1 receptors, which is the adenosine receptors. We decided that more A1 receptors being binded with adenosine would slow down the cell's activity instead of speeding it up, which would happen when we added the caffeine. And we were able to kind of formulate this hypothesis by using some work done by Brain, Bryant, and Cunningham in 2020. So the way that we decided to conduct this experiment was fairly simple. So we took our organism, which was the Daphnia magna, and we added them into a solution of caffeine that was mixed with water. It was soluble caffeine. We left them in there for 5 minutes, and then we added them to different concentrations of water mixed with adenosine. And we would measure the heart rate after leaving them in there for 5 minutes. We measured the heart rate by putting them under a microscope with a light underneath to help us view their heart a little bit easier. And then we videotaped the microscope and we rewatched it in slow motion and counted the beats for 10 seconds. And then we multiplied that by 6 for what it would essentially be in a minute. And the things that we had to keep confounding in this experiment was just the Daphnia's natural absorption of substances, such as how long it took for caffeine and adenosine to both hit their system and see the effects that it has on their system. And the one control condition that we wanted to make sure that we had was we had a Daphnia that only had caffeine. And we did this in order to assure ourselves that the caffeine actually did raise the heart rate of the Daphnia.
All right. And so the different levels that we used are right here at the top of this chart. So we used the Daphnia normal heart rate, which is just their normal heart rate, just to compare everything else from. And then we used the Daphnia heart rate plus caffeine without adenosine, which was our control for our experiment. We also used 100 milligrams of adenosine plus caffeine, 200 milligrams of adenosine plus caffeine, and 267 milligrams of adenosine plus caffeine. And these are all 100 milligrams per 100 milliliters. And so as for our caffeine dilution, we used 0.05 milligrams per 100 milliliters. And we decided on this ratio because originally we planned on putting 100 milligrams per 100 milliliters of caffeine, but we saw that that actually kills the Daphnia. So we then decreased the amount of caffeine per 100 milliliters, and we ended on this, and that's what we used. And so as for the method in which we in which we found the heart rate for each of these levels. So we began by doing this, by doing the trial systematically. So we began with all of the Daphnia normal heart rate. We went 1 through 15 and we counted that on one day or throughout many days. And then we went down the chart and we filled this out until right here. So at 100 milligrams of adenosine plus caffeine at the 5th trial of this, we changed our method a little bit after consulting Dr. Hyde. And so basically, instead of going level by level, we decided to start going, doing different levels on different days so that the slight differences of heart rate due to temperature or other things that we couldn't necessarily control that well wouldn't impact our results. All right, so as for our procedure, Walker already talked about procedure a little bit, but I just want to make it a bit more clear. So we began by taking Daphnia out and placing them into a depression slide, as you can see right here. This is the depression slide that we used to view them, but we had a bunch of other depression slides on the side with the different dilutions of caffeine or adenosine. And so we would take out the Daphnia, put them into a depression slide with caffeine, leave them for 5 minutes, and then take them out, put them, and for the control, we would leave them for 5 minutes in caffeine, for the control with caffeine, leave them 5 minutes with caffeine, then take them out, put them in a depression slide with fresh water, which is their natural habitat, for another 5 minutes, and then we would view them under the microscope. That was for the control. And then for all the other trials, all the other levels with adenosine, the only difference that we made is that instead of putting them in fresh water for 5 minutes, we put them in the different dilution of adenosine for 5 minutes. And then we placed them under the microscope and we filmed them for 10 seconds, as Walker mentioned. And then we would watch the video at home and record the heart rate into the little chart that I showed you before on Google Sheets. And so now I would like to mention another change that Walker and I made to our procedure. So we used to, like right after measuring, right after filming the Daphnia's heart, we used to count directly, like while knowing that we're doing a certain level. So if it were for the control, we knew that it was control and we were counting knowing that it was the control. We talked to Dr. Hyde and he said that that's probably not the best way to do it because we might form biases in our heads, like subconsciously, and count more or less depending on the certain dilution that we have. And so to try to fix that, after the 5th trial of the 100 milligrams of adenosine, as I showed before in the chart, we started filming the different levels and then sending them to each other without telling them what level it was. And then like that, we were able to take away that problem. All right, so this is a video of us filming the heart. As you can see right there, that right there is the heart. And so we would slow down this video and count each heartbeat. And as you can see, a 10-second video. And yeah, so that's how we did that. All right, so results and takeaways. So here's a box and whiskers plot where the box in the middle shows the middle 50% of our data. So as you can see here, the Daphnia normal heart rate is around 300. And that's the middle 50% of the data. The Daphnia heart rate with caffeine, without adenosine, our control is clearly way higher. And so that's how we knew that the dilution for caffeine that we used was effective in raising their heart rate noticeably. And so then we have the different dilutions of adenosine right here. And as you can see, in general, they're lower than the Daphnia heart rate with caffeine without adenosine. So it seems to be that the heart rate actually does go down after adding adenosine. This is a scatter plot that shows pretty much the same thing, but it just shows each individual data point. It's the same trend of, the Daphnia normal heart rate and then the Daphnia heart rate going up with caffeine without adenosine and then it going back down with adenosine in the different dilutions of adenosine. Right here, as you can see, there are two outliers where the Daphnia seem to have extremely slow heart rates and they seem to be statistically significant outliers, but nonetheless, that is why our results for the 267 milligram of adenosine plus caffeine, that level is a bit skewed. So now for the t-test. As you can see here, this is our t-test where we test the significance of our data sets compared to others. So 0.5, as you can see right there, is the benchmark which scientists use. And if the t-test gives a result lower than 0.05, then it can be said that the relationship between the two data sets is statistically significant. And we can draw conclusions from it. Conversely, if the t-test gives a number higher than 0.05, then the relationship between the two data sets is not statistically significant, and one cannot draw conclusions off of those results. So as you can see here, all of the green highlighted boxes are the ones where they do pass the t-test. And yeah, so now takeaways. So from all this data and the t-test, we can take away the fact that adenosine does in fact slow the Daphnia magna's heart rate down, which is what we hypothesized. However, we cannot make conclusions on if a different dosage or a higher dosage makes a bigger decrease in heart rate, as we also hypothesized. That came inconclusive because of the failed t-tests over here. The only one that actually did pass the t-test was the relationship between the 100 milligrams of adenosine plus caffeine and the 200 milligrams of adenosine plus caffeine. So those two data sets were statistically significant. They're different. And so there seems to be a slight raise in heart rate from the 100 to the 200. Now, we aren't sure exactly why that is, but it might be due to the fact that we changed our data collection method and our heart rate counting method halfway through the experiment. And so maybe somewhere there, there's a bit of an increase or some type of fluctuation in the data that we received. And now Walker will talk about where to proceed from here.
So there were a couple things that we discussed that we possibly could do differently if we were to do this experiment over again. There were three main things. So firstly, we would possibly redo the experiment with other methods of testing. So we might try to measure their heart rate, maybe more precisely. Like Esteban mentioned, we may do, the entire experiment with one of us having no idea which Daphnia was with which level of adenosine. And that way, maybe the results would have been different. The second way we thought about doing it differently would have been to try having a normal Daphnia, no caffeine added in the beginning, and then just add adenosine. That would have given us extra confidence that the adenosine actually does completely lower the heart rate, and we could have tried it with no stimulant added first. And thirdly, we thought about maybe giving the Daphnia a varied waiting time between doses of caffeine and adenosine. So maybe we give them, instead of 5 minutes, 10 minutes, maybe 2 minutes. We just want to switch that up and alter and see if giving it a longer time in their bloodstream would make a difference. But yeah, that's the end. Our next slide is just going to be, some sources that we used during this. And we'd like to give a special thanks to our scientist, Andrew, who helped us out a lot with this presentation. So thank you very much for watching, guys. I hope you got something out of it.